Thank you, thank you. I'm thrilled to be here. I want to thank Senator Girardi for inviting me and putting this on, and all the other organizers. I want to thank actually my prior speakers for giving a fantastic introduction to genome editing, because it sets me up perfectly to describe how we can use genome editing to cure diseases, which is something that Dr. Liu mentioned. So without further ado, I'm going to dive right in. So I'm going to start with a quote and I'm going to end with a quote. And I think these quotes actually capture not only what we as scientists like to do, but they also capture, I think, what this Congress has all been about. And so the first quote is from a technologist named K.R. Sridhar, who's actually been involved in green energy, who writes that a belief that finding innovative ways to make the world better is important. A mind in search of better ideas, even if they sound radical, is more likely to stumble across one. And I think actually sometimes the most beneficial ideas come from fields or come from areas of the world or universities that are at the outskirts of what's going on, because that is where people can think freely without being constrained by what other people have thought is how the world works. So Dr. Liu mentioned that we are not peas. We are not determined directly by our genes, and I 100% support that idea. But there are a class of diseases in which variants in single genes lead to a disease. And there are now estimated to be at least 6,000 and probably 10,000 different diseases in which a person is born with a variant in their DNA, in a gene, and because of that variant, they develop a specific disease. And these diseases cross all aspects of medicine. They include the blood system, hematology, the lungs, the immune system, the heart, the skin, the muscles, the brain, and many of you probably know these diseases. I'm a pediatric hematologist, and one of the reasons I got involved in genome editing is I wanted to figure out how we could use genome editing to cure sickle cell disease. And so that is the disease I'm going to focus on as an example of how one might use genome editing. I'll just point out that any one of these diseases is quite rare usually, but in some they affect large numbers of people, and it's estimated that about 30 million people in the US have a genetic disease, and perhaps 350 million or even more people worldwide have one of these diseases. So let me tell you a little bit about sickle cell disease or sickle cell anemia. And some people you may know it as sickle cell anemia because all patients have anemia, which means that their concentration of red blood cells in their body, in their blood, is too low. And what is the function of red blood cells? And the normal red blood cells are those round cells there. And the function of a normal red blood cell is to circulate through our blood, go to our lungs, pick up oxygen, and carry oxygen to our tissues where it can be used for all the metabolic processes that a cell needs to do. And the oxygen-carrying molecule in the red blood cell is a molecule called hemoglobin. And hemoglobin is made of two different proteins, an alpha chain and a beta chain. And in sickle cell anemia, if we look at the sequence of the beta globin gene, there is a single change from an A to a T. And what happens is that this single change in this amino acid leads to a change in an amino acid, and then that leads to the dysfunctioning of the protein, which I'll go into in a second. And it's estimated that there are approximately 500,000 people worldwide who are born with this disease per year. So if you think about it now, this is what we call an autosomal recessive disease. There's 6 billion letters in a diploid genome. Every cell has 2 copies of the genome. And these people have disease because there are 2 typographical errors. That's it. And that's what causes the disease. And as I said, the cause of this disease is a single change from an A to a T, which leads to a single amino acid change from a glutamic acid to glutamic acid to a valine. And what happens is when you have a valine in this protein, when the hemoglobin molecule loses its oxygen, the hemoglobin molecule will polymerize. It forms a stiff rod. And so instead of these molecules, these free hemoglobin molecules floating around in the cell, which allows the red blood cell to be squishy and squeeze through the small capillaries where it can deliver oxygen to the tissues, now what happens is you get these polymers, which causes the red blood cell to change its shape to look like a sickle, giving the disease its name. And these thick these stiff sickle-like red blood cells are unable to get through the small blood vessels. They form blockages, and that leads to oxygen not being able to get to the distal tissues. If you don't get oxygen to your brain, you get a stroke. And so these people have strokes, and they actually have poor intellectual development. If you don't get enough oxygen to your kidneys start to not function. If you don't get enough oxygen to your lungs, you actually start getting lung damage. And if you don't get enough oxygen to your bones, what patients with sickle cell disease have are these recurrent, what we call painful veno-occlusive crises. They get this deep bone pain that lasts for weeks. And I'm fortunate that I don't have the disease, but when a sickle cell patient described to me what this feels like, what he said is it feels like you've had your hand slammed in a car door, except instead of the pain lasting for a couple seconds or a few minutes, the pain lasts for days or weeks at a time. And I hope that gives you a sense of these recurrent painful crises that these patients are going through. So not only though, and the painting on the right was drawn by a young teenage woman who was a terrific artist who tried to draw what this pain felt like to her. Not only then are these patients going through these repeated painful crises that require strong pain medicines, is poorly controlled, but it also leads to a shortened lifespan. And if you're fortunate enough to be in the United States with sickle cell disease, you on average live to your mid-40s, which is still too short. But if you happen to be born in Africa with sickle cell disease, where most of the patients with sickle cell disease are, the average lifespan is less than 5 years of age because they don't have all the sophisticated medicines that we have in the United States. So the disease is caused by a defect in the red blood cell, which I've boxed. Now the problem is a red blood cell only lives for about 100 days. So if we could fix the red blood cell, we could maybe get about 100 days worth. But what we really need to do is provide a lifelong cure for these patients. And in order to provide corrected red blood cells, what we have to do is go back and fix what we call the stem cell, the blood stem cell, or the hematopoietic stem cell. Hematopoietic meaning blood-forming stem cell. Now, I just want to be clear here. The hematopoietic stem cell is what we call somatic stem cell. The only thing it makes is other blood cells. It's not an embryonic stem cell or a pluripotent cell, so it can't make another organism, but it's a stem cell for the blood system. And the idea is if we can fix the blood stem cell, which creates blood and red blood cells for the entirety of our life, we could cure this disease for the entirety of the patient's life. And shown in the lower left is an electron micrograph of a blood stem cell, and it just looks like a little bowling ball. So that's what it looks like. So one of the ways we can, one of the ways we can fix the blood stem cell is by doing what we call a bone marrow transplant, or a hematopoietic stem cell transplant. And in this process, what we do is we give the patient lots of chemotherapy to get rid of all their own blood, and then we take blood stem cells from someone else who doesn't have the disease, and we give them to them. And those blood stem cells from the donor engraft into the patient, and the patient will start making red blood cells that don't sickle. Now the problem is that finding a donor who's a close immune match, if we can't just do it randomly, they have to be a close immune match, is quite hard. And most patients only about 20% of patients actually have a donor. And even after undergoing what we call an allogeneic, or a transplant from someone else, these patients still suffer from a poor immune system, and they can suffer from complications of the two immune systems battling. So what we would like to do then is replace using stem cells from some random person and instead use the patient's own stem cells. Bring their own stem cells into the laboratory, fix that one base pair mutation in the beta globin gene, and then give them back. So that's the science fiction of 10 years ago, and what I'm going to show you is it's no longer science fiction. Whoops. So what we're talking about then is genome editing to correct disease-causing variants, and what we're also talking about here is personalized medicine, right? The therapy is designed for the specific DNA sequence of the patient. It's not, so we can correct a sickle cell mutation, but that's not going to be therapy for another blood disease. This is only specific for sickle cell disease. And so the way we're going to do it is by genome editing, and as Dr. Guo and Dr. Liu pointed out, the fundamental process works by designing a protein that recognizes the sequence next to where you want to fix. That protein is a nuclease. It will make a break in the DNA, and once the break is made, it activates the cell's repair machinery. The cell does not like breaks in its DNA. It's going to put that break back together. And it has two major mechanisms to put that break back together. One of which is, as Dr. Liu mentioned, is non-homologous end joining, where it's essentially a stitching mechanism where the ends get just stitched back together. And usually this is quite accurate, but occasionally what you get is insertions and deletions or mutations at the site of the break, although we don't control what those mutations are. And as the prior speakers mentioned, this is a really good way of breaking genes, but it's not a way to fix genes. And so to fix genes, we need to sort of hijack the other process that cells use to repair breaks, which is called homologous recombination. And what we do in this process is we deliver our nuclease, we make our break, and then we also deliver a piece of DNA that the homologous recombination machinery will use essentially by a copy and paste mechanism. So if I'm from California and I live right next to the Apple headquarters. So for me, it's Apple C, Apple V, right? You make a copy of the undamaged sequence and you paste it into the break and you repair the break. But by designing the donor DNA so it has that sequence change, so we can change that single nucleotide, we can trick the cell into changing the sequence of the gene. And so this is a method to fix genes. Okay, so all of those nuclease platforms work, but we have found that the CRISPR-Cas9 platform not only is the easiest, but it's the best. It gives the highest activity, and while Dr. Liu mentioned the issue of specificity, we have also found that our CRISPR-Cas9 for the beta-globin gene is highly specific. And we deliver the CRISPR-Cas9 as what's called a protein RNA complex, and that's shown on the right. So in that sort of light blue blob is the Cas9 protein, and that's going to be the part of the complex that actually makes the break. And in the yellow and then purple and red strand, that's the guide RNA that is bound to the Cas9 protein and tells the Cas9 protein where to make the break. So that 20 nucleotide sequence, that complex looks through the genome, it finds where it can match that 20 nucleotide sequence, and it triggers a series of events that leads to the Cas9 protein making a break right where you want to. Okay, so good, we can make a break. How are we going to deliver our donor DNA? Now, it turns out in the cells that I'm interested in, if you deliver naked DNA, the cells don't like that. So what we do is we deliver the donor DNA on a virus, because viruses has evolved to get into cells without the cells seeing it. And so what we do is we take our stem cells, which are those little bowling balls, we zap them with a little electricity to make a little holes in the membranes. That allows our CRISPR-Cas9 to get into the cells, and then we give the virus to allow that correction donor DNA to get into cells. Great. Does this actually work? And so what we've done is we took stem cells from sickle cell patients and we brought them into the laboratory and we did that process that I just described, and we created a donor vector that would change the T back to an A, and then we measured how frequently did we get correction. And what we found is that on average, so those 4 dots under the HR column says that in one patient we got about 40% correction, in another patient we got 45% correction, and 2 of the patients we got close to 70% correction. And what we know in this disease is that anything over 10% would probably cure the disease. Now, one of the things we have to think about is we need to preserve that stem cell property and not just correct the cells. If we correct the cells, but they're now damaged in some way and don't have the properties we want, we're not very far. So before we can take this strategy and put it into patients, we have to show that these cells aren't damaged. So what we did is we took those patient-derived corrected cells, and we put them into an animal, we used a mouse in this case, and said, do those cells continue to proliferate, survive, act like stem cells, make other blood cells? And sure enough, they do, and the correction frequency in the cells, the stem cells that engraft, is the same as the correction frequency of the cells we put in, showing that our correction process hadn't damaged those cells. So we have, and when we look at the mice, they all are healthy and everything looks good. So what that does now is set us up in 2016 to work towards a clinical trial, a first-in-human clinical trial, where we use this to try to cure patients. And I'm not going to go through all the arrows here, but what I think you should get a sense of is that this is going to be a complicated process, that it's going to take a lot of sophisticated reagents and people and facilities, but that's what we're moving to, and we're hoping to treat our first patient in 2018. I got 5 minutes. So to end, though, I want to, because as we've all discussed, that incredibly interesting audience is how, if we're good at correcting sickle cell disease, how might be applied other directions, and how would we think about which application should we do this for and which application shouldn't we do this for? And for me, and so these are 5 ethical principles that for me help me understand. Your ethical principles may be different, and one of the important aspects of deciding how we should use genome editing is to make sure we get lots of people's input. So the 5 principles for me is, one is based on John Rawls' Veil of Ignorance. And what he wrote is that we should design a world in which we don't know where into that world we will be born. So for example, well, I'll get to this, but if I might be born into the world in Africa with sickle cell disease, I might be born into the world in Palo Alto with sickle cell disease. I want to create a world where it doesn't matter where I was born. I still have access to the same therapy. I want to make sure we minimize suffering. Earlier in the morning, somebody talked about utilitarianism and maximizing benefit. I don't know what benefit is, but as an MD, I know what suffering is, and I want to minimize suffering. I want to minimize structural inequality. Again, it goes back to the John Rawls. I don't want people to be born where there's inequalities that are baked into our world. I want to make sure we don't treat individuals as objects. Individuals are human beings. They deserve to be treated as human beings. We should not be designing people to perform certain tasks. And I think we need to have humility to recognize how much we don't currently understand, and to make sure that if we do something and there's unanticipated consequences, we recognize the risks we've taken. So in terms of distribution, I think one of the really important issues is to make sure in the next 10 or 15 or 20 years that we can take a process that I can do in my hospital at Stanford, California, and bring it to the entire world, including to Africa. And how do we do that? I'm not sure, but there's lots of smart people in here, lots of passionate people, and hopefully we can be partners in that. And then the last ethical thing I want to talk about is the various ways one might use genome editing. And this is a box that came from a 2x2 box that came 40 years ago, as people were talking about whether you should manipulate the DNA to cure disease, or should we manipulate DNA for enhancement to improve traits that's not related to disease. Should we do that in somatic cells that only are in the patient, or should we do it in the germline, which would allow the change to be passed along to future generations? And so in the top left, I put in green because I think using genome editing to cure disease is something we should pursue. It minimizes suffering. It would be a world in which I would want to be part of. On the other hand, I think that using somatic cell editing for enhancement, and as I agree with Dr. Liu, exactly how to do that I don't think is going to be very easy, but I put that in a light pink because I think that that would be a way of creating more inequality in the world. And I would not want to be born into a world where some people had access to being able to be enhanced and other people didn't. I'm a little different than others in which I think that perhaps for certain diseases under well-defined regulatory regimes that can't be cured by somatic cell editing, we may actually have to think about editing such that that change would be passed along to future generations. But that's obviously a conversation that we have to have broadly, because once we do it in one country, it can spread to other countries. And obviously in bright red, I have that the concept of using genome editing for enhancement is something we shouldn't do, although there are philosophers out there, that's the transhumanists, who say that humans are fundamentally flawed and we should do everything we can to improve those flaws. So with that, I want to end with a quote, which is to say, I want to stay as close to the edge as I can without going over. Out on the edge, you can see all kinds of things you can't see from the center. And again, that goes to the point that when you're on the edge, you can actually see things that you can't see in the middle, and you should take advantage of that here in Chile. And in honor of the painting and mural above my head, I'll close with a picture of my dog, and I think the panel will now come out. So thank you very much.